Hi, my name is Lauren Banks, and I'm a PhD candidate in the Streams Lab at the University of Waterloo. And today I'll be talking about part of my PhD research, which is looking at reach-scale groundwater inputs as a driver of spatial patterns in ecological structure and function. Before I get into my research, I want to talk about groundwater contributions to streams. Groundwater fluxes in streams are areas of downwelling, where the surface water enters the groundwater, and upwelling, where the groundwater enters the surface water. In upwelling areas, there are typically more moderated temperatures, additional nutrients, and this helps maintain stream flow. Groundwater fluxes have been shown to have impacts on stream ecology, where areas of upwelling typically have higher benthic algal biomass, which is generally associated with these additional nutrients and more moderated temperatures compared to downwelling areas. However, previous work has focused on streams with low productivity and primarily in riffle habitats. In agricultural streams, typically there is high nutrients and limited riparian cover. Like all streams, there are sections of runs, shown here by these straight lines, that tend to be a little bit deeper and have slower stream velocities than riffles, shown here with these squiggly lines that tend to have faster stream velocity and are generally a bit more shallow. Now I want to draw your attention to the bottom of the stream where we have the stream bed and this hyporheic zone where groundwater and surface water exchanges. The stream bed in the hyporheic zone is teeming with life and generally dominated by invertebrates and microbes. The microbial community can form a scum on rocks or other sediment, and we more affectionately call these biofilm, which are made up of bacteria, fungi, algae, diatoms, and viruses. Biofilm make up the base of the food web, and therefore understanding what influences patterns in biofilm structure and function is imperative for ecological management. My study has two questions. The first, does stream biofilm structure and function differ based on the amount of groundwater input? Here we hypothesize that resource subsidies provided by groundwater input will alter stream biofilm structure and function. The second being, are these differences dependent on habitat type, so whether you're in a riffle or a run? Here we hypothesize that based on differences inherent to runs and riffles, namely velocity, will impact how groundwater subsidies are taken up by stream biofilm, and that this will ultimately affect stream biofilm structure and function. Our study took place in Kintore Creek in Ontario, Canada. Land use in the catchments is over 80% agriculture, which we can see is denoted by this light green color here, followed by forested areas with this dark green color, and urban and residential uses in the pink color. There's typically high surface water nutrients across the catchments with variable groundwater input and open canopies. So my work has included studies across seasons at the stream segment scale and at the microhabitat scale, but today I'll be talking about my work at the reach scale. For my field study, we selected 3 reaches in Kintore Creek, 2 with high groundwater input and 1 with low groundwater input. And here shows a schematic of our field study where we had selected 2 riffles and 2 runs in each habitat and placed 5 artificial substrates with tiles for biofilm to grow and used a standardized method called the cotton strip assay to measure organic matter processing. Take a closer look at one substrate here on the bottom left. Biofilm grows on the tiles, and each of the 3 tiles is analyzed for chlorophyll A accumulation, biofilm growth rate, and diatom taxonomy. We use the cotton strip assay to measure streambed and subsurface organic matter processing. Here we had our streambed cotton strip assay attached to our artificial substrate, and we buried the subsurface cotton strip assay 10 centimeters into the streambed. We also measured the stream environment, which included water chemistry, temperature, nutrients, as well as stream physical characteristics. We also use groundwater proxies, including radon 222, vertical hydraulic gradient, which is a measure of the direction of water movement, so whether it's coming up from the groundwater or entering the groundwater, stream bed temperature, and substrate type. So let's get into some results. Here we have the reaches divided by groundwater input as high groundwater reach 1 or HG1, high groundwater reach 2 or HG2, and our low groundwater reach, LG. Each reach is further subdivided on the x-axis into the 2 habitats, riffle and run. And on our first panel, we have chlorophyll A accumulation on the y-axis. And on the second panel, we have biofilm growth rate on the y-axis. We can see that runs in the high groundwater reaches have greater chlorophyll A and biofilm growth rate compared to the low groundwater reach, and significance here is denoted by these stars. Here we have the results of our partially square analysis that produce one significant axis. Here, triangles represent runs and our colored circles represent riffles. We can see that chlorophyll A and biofilm growth rate is positively associated with depth and negatively associated with stream velocity. And this is a pattern that we would expect from shear stress resource subsidy trade-offs. We also see that habitats within the reaches generally cluster together, with the exception of HG1, where the riffles show quite a bit of spread. Now on to my favorite creatures, the diatoms. So here, 6 taxa accounted for over 80% of the total relative abundance across all of the habitats. So similar to our chlorophyll A and biofilm growth rate figures, we have the reaches divided up by groundwater input and further subdivided on the x-axis into our 2 habitats. Here we have total relative abundance of diatoms on the y-axis. Here we can see that our high groundwater reaches are typically dominated by Coccinase placentula, which is an epiphytic diatom, whereas for our low groundwater reach is generally more dominated by Achnanthidium eutrophilium, which is a pioneer species. Additionally, diatom community composition in riffles and runs in the high groundwater reaches were significantly different, again denoted here by stars, a pattern that we did not see in the low groundwater reach. Our analysis suggested that these patterns were associated with depth and pH. However, depth and pH are not all that variable among the reaches and largely tracked with existing reach characteristics rather than with groundwater input. Here we have the results of our cotton strip assay for the stream bed. Here we measured organic matter processing as percent tensile loss per day. On the figure, once again, we have our reaches divided by groundwater input and further subdivided on the x-axis into our 2 habitats. Here on the y-axis, we have stream bed percent tensile loss per day. Similar to chlorophyll A and growth rate, we found that rates of tensile loss at the stream bed differed between habitats, again, only in the high groundwater reaches, where the stars here denote significance. Partially square analysis for percent tensile loss at the stream bed produce one significant axis. Again, here we have triangles representing runs and our colored circles representing riffles. We see that stream bed percent tensile loss is positively associated with stream velocity and water temperature, a trend found by other researchers and is generally attributed to greater physical abrasion with faster stream velocity and higher temperatures stimulating microbial activity. Additionally, habitats and reaches tended to cluster, with riffles in our high groundwater reach 1 tended to show, again, the greatest spread. However, differences in water temperature among reaches and among habitats was low and did not produce the pattern that we would expect with groundwater input, that is, cooler and more moderated water temperature. Now on to percent tensile loss in the subsurface. First, we can see that generally rates of tensile loss are faster at the stream bed than in the subsurface. And this is a pattern that's consistent with past studies and likely due to less physical abrasion and limited access by microbial and invertebrate decomposers in the subsurface. So focusing on the subsurface, again, we have our 3 reaches divided by groundwater input and further subdivided on the x-axis into our 2 habitats with subsurface percent tensile loss on the y-axis. We found that groundwater input in the reach was driving differences in percent tensile loss with no effective habitat. This is in line with past work where conditions in the hyperrheic zone was the primary driver of substrate tensile loss. Here, our high groundwater reach 2 had significantly faster rates than high groundwater reach 1 and our low groundwater reach, again, significance denoted by this star. And this is likely due to differences in the hyperrheic exchange of dissolved oxygen, nutrients, and chemical constituents. So taken together, what did we learn? Firstly, that groundwater matters. We found that stream biofilm structure and function did respond to differences in groundwater input, but only in runs. We didn't really find any compelling evidence that groundwater input altered the environment to the extent that we would expect a biological response. It appears that at this scale, high surface water nutrient availability and the dilution of localized thermal effects by surface water may have overwhelmed any biologically meaningful differences generated by environmental variation in groundwater inputs at this reach scale. This suggests that perhaps the effects of groundwater input are more difficult to detect in high nutrient streams at the scale that we measured groundwater input, that is, at the reach scale. The differences we found in stream biofilm structure and function among habitats were consistent with past work examining among habitat differences in biofilm biomass and organic matter processing, where runs tend to accumulate more biofilm biomass and riffles tend to have faster rates of organic matter processing. The limited variability of measured environmental conditions that we observed may be due to a scale mismatch of measures of groundwater input, which we did at the reach scale, and the local effect of groundwater, which is more at the point or microhabitat scale. Therefore, when considering the role of groundwater input in generating ecological heterogeneity, future work must consider both the context, so the stream characteristics, and the scale of measurement of groundwater, looking at specific points of groundwater flux versus more coarse measurements of groundwater input at the reach scale. I want to thank members of the Stream Lab who helped me out in the lab and in the field, as well as our collaborators at Environment Canada and in the Restore Lab at Western University, who conducted the groundwater research for this work. I want to thank NSERC for our funding, and if you have any questions, you can contact me via email or you can find me on Twitter. Thanks for taking the time to watch my recorded presentation.